Welcome. In this video, we look at experiments and testing, the tools we use to validate ideas and solutions. Experimentation is the backbone of scientific testing. It allows us to test hypotheses under controlled conditions. To do this effectively, we rely on key concepts like decisive test, causation, underlying process, and bias. In order to move from hypothesis to a tested explanation, we need to test an experiment that creates artificial and controlled circumstances. Let's look at the example of Francesco Redi. In the 17th century, he wanted to challenge a doctrine of his time, spontaneous generation. This theory speculated that some active principle would be necessary for spontaneous generation. Life, example oysters, scallops, and limpets, could arise from inanimate matter such as dust, or that maggots could arise from dead flesh. The doctrine goes back to the Greek philosopher and naturalist Aristotle and was taken as a scientific fact for 2 millennia. Aristotle never accepted experiments as a scientific method and was rather relying on logical reasoning. Francesco instead conducted an experiment. He put meat into jars, one open, one sealed, and one covered with fine Naples peel that kept flies from coming into contact with the meat but did allow air to circulate. Carrying out this experiment, Redi obtained the expected results. Maggots appeared only in the uncovered jars. By this approach, Redi was able to rule out the possibility that something in the air might be responsible for his results. As a result, the conclusion that fly droppings were responsible for the maggots was on a much stronger footing. For building a good test, you need to fulfill 2 criteria. The first is falsifiability, the idea that your experiment must be able to prove your hypothesis wrong if the evidence doesn't support it. Redi's hypothesis was simple. Maggots come from flies, not from spontaneous generation. It could have been falsified if maggots had appeared in the sealed jar, where no flies could reach the meat. But that's not what happened. Maggots appeared only in the jars where flies had access to the meat, supporting his hypothesis rather than disproving it. Because his design allowed for possibility of being proven wrong, his findings carried stronger scientific evidence. The second criterion is verifiability, ensuring that there's no other reason for the observed result. Redi achieved this by using Naples paper that let air in but kept flies out, proving maggots were not coming from the air itself. His experiment was verifiable because it met 3 conditions. Controlled condition, variables like fly exposure were carefully managed. Reproducibility, other scientists could repeat it and get the same outcome. Empirical evidence, conclusions were grounded in direct observation and measurable results. This allowed others to independently confirm his findings, strengthening the rejection of spontaneous generation and supporting biogenesis, the idea that life comes from pre-existing life. In an experiment, the independent variable is what you change or control to see its effect. In Redi's case, it was the presence or absence of flies, which he moderated by covering the jars. The dependent variable is what you observe and what you measure. Here was the appearance of maggots. Redi's setup included both experimental groups, open jars, and control groups, sealed jars. This design allowed for both repeatability, where the same researcher gets the same results, and reproducibility, where other researchers can confirm the findings. More generally, a well-designed experiment isolates prediction that will happen if an explanation is correct. This means arranging the right circumstances, either in controlled lab conditions like Redi's fly jars, or in the real world, such as studying historical data under different conditions. Once the setup is in place, we compare the prediction to the actual facts. The prediction must be capable of rejecting the explanation if it's wrong and confirming if it's correct. That means ruling out factors that could cause predictive failure in the explanation were true. This is falsifiability. And ruling out factors that could cause predictive success even if the explanation were false. This is verifiability. Depending on what we find, the experiment may need to be refined for stronger, more decisive results. There may be modifications needed in the experiment design. Example, Reddy's third jar covered by Naples paper, letting air circulate but keeping flies away. Following this process, you create a well-designed experiment. To establish a causal link, we begin with a null hypothesis, the assumption that there's no effect at all. In Reddy's case, that would mean maggots appear in any jar, regardless of whether it's covered or open. The first step is to reject this null hypothesis by showing that there is an observable difference. Next comes the question of statistical significance. How much does the effect differ between the experimental group and the control group? For Reddy, that meant comparing the number of maggots in open jars versus covered jars. The term statistical significance has a precise meaning in science. In formal research, we only use it when we have actual statistic evidence, typically at the 0.05 level. This means that there is greater than a 95% probability that the difference between groups, such as open and covered jars, is real and not due to random chance. As you have seen before, theories emerge from integration of multiple tested hypotheses. In this example, you can see how content type, media type, and posting time influence user engagement on Facebook, measured through likes, comments, shares, and interaction duration. By experimentally testing each of these factors, you can understand how the results combine into a broader perspective, giving you theoretical insights into the mechanisms that drive engagement on the Facebook platform. The mechanism behind describing relationships is the mathematical concept of correlation between two sets of values. Correlations can be positive, negative, or none. Two factors are positively correlated if a higher percentage of people with characteristic P also have Q. They are negatively correlated if a smaller percentage of people with P have Q. And they are not correlated if there's no meaningful difference in Q between those with P and those without. The key point you should remember, correlation is not causation. Assuming an individual relationship from a correlation means assuming a causal link. And that's a leap we can't take without further evidence. Suppose a study finds that projects with more GitHub commits tend to have fewer critical bugs. It might be tempting to conclude that just increasing the commit frequency will improve software quality. But the real cause could be something else. For example, more experienced development teams commit more often and produce higher quality code. The commit count and software quality are correlated, but one does not directly cause the other. Correlations may arise from genuine cause, from coincidence, or from some other underlying factor. To truly understand the connection, we must investigate the underlying process. An underlying process means explaining something by looking at what is really happening inside. In a reductionistic approach, you take what you see and break it down into smaller, more basic parts. For example, in the 18th century, Vaucanson's automatic duck looked like it could eat and digest food. The duck had over 400 moving parts in each wing alone and could flap its wings, drink water, and digest grain. This composition of components helped to explain the underlying biological process within ducks. The same idea appears in computer science. For instance, look at the paper of Sergey Brin and Lawrence Page, the founders of Google, where they describe the architectural concept. A search engine feels like it understands your query, but under the hood, it's a combination of index structures, ranking algorithms, and statistical models, not comprehension in human sense. In both cases, what you observe at the surface can be fully explained by the underlying process, which may follow deterministic rules or statistical laws. We will revisit the concept of understanding the underlying process by building artifacts when we talk about design science. In any good experiment, you need to ask a few key questions. Have you controlled for all possible causal factors other than the one you're testing? Could experimenter bias shape the outcome? Could the expectations of participants change how they interact? And finally, is any difference you see actually statistically significant for your sample size? Let's get back to our search engine example from the previous slide. Testing the efficiency of search algorithms, you need to make sure that only the ranking algorithm is different between the systems you're comparing, and everything else stays the same. Example, same data set, identical hardware in network conditions. If you don't control these factors, any observed improvement could be due to something else, not the algorithm itself. If you let only your own team test it, their familiarity with the project could bias the results. If testers know which system is the new one, they might favor it simply because they expect it to be better. And if you don't run proper statistical tests, you might think the new algorithm is an improvement when it's actually just a random variation. A strong experiment rules out other explanations, keeps both participants and experimenters from influencing the results, and use statistics to confirm that the effect is real. You have seen the core building blocks of a sound experiment for testing explanations. You have learned about falsifiability and verifiability, designing experiments that can prove an idea wrong and confirm it if it's right. We looked at independent and dependent variables, what you change and what you measure. We explored how a decisive test can isolate the key prediction and rule out other explanations. From there, you understood to distinguish causality from correlation, learning why a relationship between two things doesn't always mean one causes the other. We uncovered the underlying process, explaining phenomena by looking at what's happening at a deeper level. And we ended with bias, recognizing the need to control for other factors, avoid influencing results, and confirm findings with solid statistics. Together, these principles form the foundation of designing experiments that are clear, repeatable, and conclusive.